Okay, so bakit naka-expose sa outer at inner part ng cell yung hydrophilic na end, which is yung water-loving end, and bakit nakatago yung water-fearing o yung hydrophobic end doon sa insides ng bilayer natin, no? So basically parang you want to preserve that form, that organization because ma mag kumbaga pag one is exposed, mas magkakaroon ng chances na siya ay maging ma-dissolve within dun sa solution na kung nasan man yung cell. Okay? But sometimes we can actually dissolve it manually, di ba? Kaya pag sa DNA extraction, one of the first things to do after maceration or parang after sirain yung sample nyo is i-introduce nyo siya sa detergent, which will serve as I forgot the term. Huh? Emulsifier, yeah, I think that's the term. Emulsifier para makuha niya yung mga fats or phospholipids in this sense para matanggal niya yung phospholipid bilayer detergent yung ginagamit. Okay? So it's composed of two fatty acids, fatty acid tails. So yung mga parang legs niya, yun yung fatty acids attached to a glycerol. Okay? The third hydroxyl joint to phosphate group anyway. An additional small, wag na rin yan. Okay, so here is a phospholipid. Like I said, it's composed of two fatty acid tails. Yung dalawa niyang parang paa are two fatty acids. And these are the hydrophobic ends, diba? The hydrophobic tails. And if you notice, one of the legs o yung isa sa mga tail natin ay hindi siya naka-straight talaga. May bend. And this is due to a cis double bond. So merong cis double bond na nag-exist dun sa isa sa mga tails which creates this kink, yung term niya, kink o yung pagka parang bali niya ng onte. Okay? So ito yung tinutukoy na phosphate. So this is the glycerol. Okay, this is the glycerol and this is the phosphate kung saan siya naka-attach. So sometimes daw merong mga attachments like choline, but sometimes hindi siya choline. Kumbaga this will serve like as the R group. Nagva-vary siya dun sa phospholipid natin, if I'm not mistaken. Okay, so this is the phospholipid. Again, it's a bilayer in the cell membrane and it's the main component of the cell membrane. Okay, siya yung bread and butter ng cell membrane because without the phospholipid, the cell membrane would cease to exist. So that this structure itself, yan yung nagper-permit na magpapasok ng mga ilang bagay as well as magpalabas ng mga certain na bagay din. Okay? So, it all depends sometimes on the size, the charge of the compound or the molecule, and such. Okay. Okay, so here is the actual cell membrane, the phospholipid bilayer. So, if you notice, there are also cholesterols attached to, ay, hindi kita. There are also cholesterols attached to the cell membrane itself. Okay? So, may mga cholesterol na naka-attach dyan. That will serve as, ang function niya is to like bind the phospholipids, kumbaga parang also to keep it together, but at the same time, it keeps it fluid. Kasi our cell membrane is not a static structure. Hindi siya yung kung ano isipin natin na parang cell wall, kunyari, di ba? Hindi siya static na structure. Okay. Okay na. Thank you. So, hindi siya static na structure. More on fluid siya. That's why the model ng cell membrane natin, ang tawag is fluid mosaic model. Kasi nagfo-flow siya. Okay? Nagfo-flow siya. Yung phospholipids natin, hindi siya static. Gumagalaw-galaw sila. Okay? Kumbaga parang they're free-floating dun sa organization nila. That's why sometimes may mga cases na yung phospholipid na isa, pwede siyang lumipat dito sa kabila. Tapos, vice versa, may lilipat din kunyari sa taas. Pwede silang mag-flipan. Okay? So anyway, so yon fluid talaga yung ating cell membrane. Yung phospholipid bilayer natin, fluid siya. Okay? And yung fluidity niya, it partially owes this fluidity to the cholesterol found in the cell membrane. So, hindi naman all bad ang cholesterol. Okay? So, may natural purpose siya. For example, here in the cell membrane, its natural purpose is to keep it fluid as well as to bind together the phospholipids. Okay? So we have several proteins found in our cell membrane. Kapag po yung protein ay siya ay kumbaga tumatagos dun sa dalawang bilayer, nako-cover niya yung dalawang bilayer like this one and this one, this is what we term as integral proteins. Integral. Okay? So, these integral proteins, usually, sila yung mga nagsiserve as channels or sometimes they serve as yung primary na pang-transport, okay? A protein transport for certain molecules and compounds. Like I said, kasi di ba, hindi naman readily nakakapasok yung certain compounds or certain molecules papunta sa cell. Okay? Ganun din palabas, hindi din kagad-kagad nakakalabas. So yung channel, okay, yung mga protein channels o yung mga pang-protein transport natin, sila yung nagsiserve as parang gateway or di kaya sila yung nagsiserve bilang way para makapasok yung mga compounds na yun o yung mga molecules na yun papaloob or pwede din naman papalabas ng cell. So sila yung gumagawa ng paraan for those molecules. Okay, so pwedeng iba yung charge ng protein na to kaya mas madali makapasok yung certain na molecule. Pwede din naman meron siyang certain na shape or certain configuration para ang i-allow lang is yung certain na molecule na yon with this specific structure. Pwede pong ganun yung case niya. Okay, so again, these are integral proteins. Kailangan sila ay nag-persist doon sa bilayer. Kailangan tagos siya doon sa dalawang bilayer. Paano naman sir kapag nandito lang sa taas lang or parang ganito lang, nandito lang siya sa baba? No? So like this one then. These are what we call peripheral proteins. Okay? Peripheral proteins, these are more on cell receptors. They are more on what we call cell receptors. Sir, what are cell receptors? Basically, ginagamit siya ng cells, ginagamit siya ng cells pangkilala. So, para makilala ng cell kung, let's say, may interaction siya with another cell. Okay? This is more specific to immune cells, yung mga ganyan, no? So, these cell receptors will serve as like a guide para ma-identify nila kung ano yung cell na yun or not. Okay? Sometimes then, these cell receptors, they can be used to like, ano ba to? Receive a message or receive a signal. Okay? So, kunyari, specific, di ba? Si peripheral protein o si cell receptor natin. So, kung ano man yung specific na signal na fit doon sa receptor natin, yun lang yung tatanggapin ng receptor. So, kung meron man produce yung katawan na yung specific na signal na yun at nailagay siya doon sa cell receptor natin na yun, sa cell, malalaman ng cell kung anong dapat gawin. Kasi kung para saan ba yung signal na yun. For example, signal siya for apoptosis or yung program cell death. Okay, so pwedeng ganon. Sige, tignan natin dito yung example no, sa cell receptor. Yung sa cell receptor sa HIV, for example, the HIV utilizes one of our cell receptors in our immune cells. So, isa sa mga immune cells natin, I think it's called CD4 na, meron siyang tinatawag na CD4 receptor, yung cell natin na yon, yung immune cell. So, si HIV, ni-utilize niya yung CD4 receptor para siya ay makapasok papaloob dun sa ating immune cell. Okay? So, ginagawa niya is, pag nag-attach na yung virus dun sa cell receptor, sa CD4 cell receptor, mag-open up ngayon yung cell, i-receive niya yung genetic material ng virus. So, basically, parang hina-hijack niya yung receptor. So, pwedeng ganun. Pwedeng mangyari yon and I think, yeah, evolution paved its way na talagang magkaroon ng mga ganong chances, no? Sa bacteria naman din, ganun yung cases, okay? Sometimes, there are receptors cell membrane of the bacteria which allows certain viruses or pwede din like certain compounds or molecules na maipasok dun sa loob ng bacteria. Okay, so parang ganun yung idea. Pero yun nga, hindi lang naman siya for infectivity. It can also function for like bodily signals, ganyan. So for example, nag-produce tayo ng um, ano ba yung pangalan nun? Basta parang simulating hormone sa ating mga gametes, okay, sa ating mga sex organs. So pag na-receive yun ng mga cells natin sa ating sex organs, it will be signaled to, let's say, produce egg cell or produce a sperm cell. So parang ganun yung idea. Okay? Wait. Okay. Okay, so cell transport. So, na-discuss na natin si plasma membrane and some components of the plasma membrane. Let's move to the cell transport, which is yung kasaklaw na topic ng plasma membrane, no? So, kung si plasma membrane, like I said, it regulates the entrance and exit of certain molecules and compounds. Basically, it's responsible for cell transport. It's the primary structure responsible for cell transport because it's the one regulating the entry and exit of molecules and compounds. So we have two types of cell transport, active and passive transport. Okay, so basically the only difference here is that for active, you use energy and for passive, you do not use energy. Okay, so for passive transport, it usually utilizes the gradient. Okay, kung ano man yung gradient natin, for example, 'di ba, from high concentration to low concentration, laging ganun yung movement, 'di ba? So, yung passive transport, lagi niyang utilize yung gradient na yon, which is from high to low concentration. Lagi niyang ginagamit yon. Si active transport, pag against ka dun sa gradient, usually active transport ang kailangan mong gamitin. Okay? So this is with the case of sodium potassium pump. So like I said last time sa nervous tissue, the sodium potassium pump, siya yung nagbibigay ng um, unbalanced na number of ions sa loob at labas ng cell. So gusto niya laging unbalanced yun because sa nervous tissue, kailangan niya nung differences nung ions para mag-produce nung ating charge o nung ating impulse, which will be the signal for our nerve cells, di ba? So basically, yung differences sa ion na yon, nangyayari yon because our nervous tissue have sodium potassium pumps in their membranes. So yung ginagawa ng sodium potassium pump, gumagamit siya ng energy to keep the ions unbalanced. Kailangan unbalanced siya. So ganun yung nangyayari sa active transport. For passive transport, we have I think 3 types. Ayan. So for active transport, gagamit siya ng energy in the form of always, 'di ba? ATP. Okay, so gagamit siya ng energy in the form of ATP to keep the molecules or to force the molecules against the concentration gradient. Pero pwede naman ding hindi against, but most of the time, active transport is used for transporting molecules, compounds, or ions against the concentration gradient. So for example, mataas yung concentration dito nung diamond, gusto natin unbalanced siya. So mag-pump tayo ng mag-pump papalabas ng diamond doon sa extracellular environment para unbalanced sila. So dito mataas yung diamond na concentration, dito mababa. So unbalanced. So for example, yung circle naman dito sa labas mababa. Tapos yung circle dun sa loob ay mataas. Okay? Tapos kunyari magkaiba pa sila ng charges. So you will create an environment that is, for example, inside the cell more negatively charged and then outside the cell more positively charged. So sa nervous tissue, this is the exact example. Yan yung gusto niyang mangyari na unbalance in charges to create again for it to be able to create an impulse or a charge. Kasi 'di ba yun yung signal natin sa nervous tissue. Okay, so for passive transport, we have 3 types. Diffusion, osmosis, and facilitated diffusion. Basically kasi, Yung osmosis kasi parang given na siya that the movement of water in and out of the cell based on solute concentration o yung concentration ng solute. So si water daw, mag-move siya papaloob ng cell or papalabas ng cell depende dun sa solute concentration. So for example, mataas yung solute sa labas ng cell. Water will then move out. Gusto niya kasi mabalance eh. And then, if naman mataas yung solute sa loob, water will move in. Ganon, para to keep it balanced lang. Okay? So, diffusion simply, wait, meron pala tayong definition. Yan. So, simple diffusion, movement of small molecules, mostly gas molecules. Literal na nagde-diffuse lang yung ating mga small molecules into and out of the cell. So, fina-follow niya din yung concentration gradient. Pero wala na tayo masyadong case dito because tumatagos lang naman siya sa phospholipid bilayer natin pag diffusion. Okay, pag diffusion, tumatagos lang sa phospholipid bilayer. So, wala tayong masyadong issue doon. So, ito naman, for example, osmosis. The movement of water is controlled by a protein channel. Okay, it's controlled by a protein channel called aquaporin. So, it's a protein channel and aquaporin is responsible for moving water in or out of the cell. Para siyang, pag protein channel, para lang siyang highway. Okay, so kung notice nyo, wait lang, aquaporin, no, wait. Kung notice nyo, pag protein channel siya, diretso lang, highway lang siya. Pero ito, may mga protein transport tayo na hindi siya highway. Para siyang nag-open in and out. Ganun siya. Pwedeng ganun. Okay? Pero pag sinabing protein channel, diretso lang yung protein o yung molecule, diretso lang siyang tatagos dun sa protein. Okay? So osmosis again, it's controlled. The movement of osmosis is facilitated by a protein channel called aquaporin. Okay, so the movement of water again is based on the solute concentration in and out of the cell. Saan ba mas mataas yung solute, doon mag-move yung water. So kung sa labas mataas yung solute, papalabas yung movement ng water. If sa loob yung taas ng solute, papaloob yung movement ng water. Facilitated diffusion, basically, kapag siya ay gumagamit nung channel o nung protein transport, yung nag-open in and out, that is termed as facilitated diffusion. That's why dito, di ba, kung mapapansin nyo, yung protein channel, tsaka yung pang protein transport natin, they are both considered as facilitated diffusion. Okay? Yung osmosis, completely different thing siya. So, huwag nyo siya isasama dito sa facilitated diffusion. Although, technically speaking, facilitated siya ng protein, iba pa rin yung movement ng water compared sa ibang molecules or compounds natin. Okay? Kaya, separate siya dun sa passive transport natin. Yan. So, for the movement of water or for osmosis, if you can see, the plant and animal cell has different normal states. Sa isotonic solution, dun yung normal ng animal. Pero sa hypotonic solution, dun yung normal ng plant cell. Kasi ang plant cell, lagi siyang turgid. So that's the normal state of plant cell. Gusto niya madaming water sa loob niya because the vacuole will hold much water to keep turgor pressure. Kailangan parang pressured kasi lagi yung cell ng plants. Okay? So, pag naman sa hypotonic solution, yung cell ng animals, naglalise siya or nabubutas siya or nagbo-burst siya. Bakit? Because the solute concentration is higher inside. That's why water will move inside the cell. Tapos, pag nag-move na move inside, tumataba sa cell, possible siya na ay mag-burst o mag-lyse. Because hindi niya kaya i-accommodate yung volume. Okay? So, mataas na solute concentration. Sa loob, hypotonic solution. Isotonic, basically, normal. So, tama lang yung number na solute concentration in and out of the cell. That's why yung movement ng water, sakto lang papaloob, sakto lang din papalabas. Pag hypertonic solution naman, mas mataas yung concentration ng solute sa labas ng cell. Okay? So dahil mas mataas yung concentration ng solute sa labas ng cell, magmo-move ngayon yung water papalabas ng cell, making it shriveled or plasmolized. Okay? Plasmolized. Plasmolized, basically speaking, sa plant cells, kung mapapansin nyo, yung cell membrane, hindi na siya dikit doon sa cell wall. So, yan yung palatandaan na siya ay nag-plasmolize na because yung water movement ay papalabas na nung cell. So, hindi na turgid yung plant natin. Okay? Ayan. I think that is all. Do you have any questions?